Thank you. Good morning. My name is Dave Bassard. Jason Lambert to my right and I will do the initial presentation. But the work we're talking about has been done by a much broader group of people. So we've got a panel who can help answer questions and take it wherever the committee wants to take it as we go. So let me introduce Dr. Jason Lambert to my right, Ted Berner, Alan Davis, Jeff Gift, Karen Hogan, Leonid Kopilov, and Ravi Subramanian.
Leonid's not there.
What?
Leonid's not up there.
Where's Leonid?
It's virtual.
An approximate Leonid. Thank you. Glad to be here. As we go through this, I'm going to focus on the key recommendations that were in Table 8, but we're also going to take the opportunity to talk about other things that we're doing that relate to those topics as we go through. Let's see. So there were 3 priority recommendations in the 2014 report that relate to quantification. One was with toxicity values, that EPA should develop criteria for determining when evidence is sufficient to derive toxicity values. And we'll talk about how we're using the hazard conclusions to do that. The second, points of departure, that EPA should clearly present 2 dose response estimates, a central and a lower bound. And we'll discuss that we do that now in our documents. And quantitative capabilities, that EPA should expand its ability to perform quantitative modeling of evidence integration, in particular, develop the capacity to do Bayesian modeling, and emphasize that iris assessment should not be delayed while this capacity is being developed. So we'll talk about some of the things we've done, the evolution that's going on, and recognizing that that's something that we're continuing to learn about and continuing to develop. So the first of the recommendations, evidence integration conclusions inform when to develop toxicity values. So there was a fair amount of discussion yesterday about the structured process that the IRIS program uses to reach summary conclusions. And we mentioned that the exact phrasing of those conclusions is an active topic within EPA in terms of how to best communicate them. But we think it's pretty stable that we've got this process of reaching them, and we're debating what labels to use more than how to get there. And much as in the cancer guidelines, our handbook talks about in the first 2 levels of evidence, here's Leonid Kopilov, that we will derive quantitative toxicity values where the data allows us. In the weaker conclusion, it really depends on the situation, that part of problem scoping and formulation could be talking with programs, talking with clients to understand if there's a situation. They may, for example, want a number for screening purposes or priority setting purposes. So even though the hazard conclusion may not be as strong as in the top 2 categories, there may be an interest in a number with the appropriate caveats and considerations around the hazard conclusion. There nonetheless might be advantages for screening or priority setting. So we think we've met that first recommendation from the committee. We're aware that we also need, as we go forward, to figure out which studies we take into dose response. So there was a fair amount of discussion yesterday about inclusion-exclusion criteria, how we evaluate studies. And of course, many of those same considerations about study quality are also considerations in terms of whether a study is useful for quantification. But when you get to dose response, there are additional considerations. So for example, you could have an epidemiology study that's quite good at saying the exposed versus the unexposed. But if the characterization of exposure is not good enough to put an actual dose or concentration to that, it's not going to be nearly as useful for dose response as it might have been for hazard ID. So there are additional considerations that come in as we do this. At this point, we think that's still a matter of expert judgment, that it's not a simple formula. but that there are some, much as yesterday's discussion, there are some standard questions, some standard considerations to go through. And then, because each of the data sets are often quite different, then there's a responsibility on our side to then articulate carefully as we go through an assessment what were the factors we used, how did we make those judgments given the set of studies that was available to us. And this is a screenshot from work we've done for the handbook, just going through some of the considerations. So as you can see, if we have a good set of human epidemiology studies, that's going to get our primary weight. And there may be some cases where part of the modularity decisions early on could be that if there's a lot of epidemiology data, we may actually focus on the epidemiology data for that assessment. Where there's a mix, we'll look at both, but we generally have a preference, given we're trying to estimate the risks to humans, to use the human data and give it the most weight if it's good studies and can be used for dose response. With animals, there are additional considerations, whether the animals are a good model for human responses. And you can read down through there things like if we're trying to get an inhalation value, we prefer having an inhalation study. If not, we have to use PBPK modeling or other considerations if we want to go there. So moving on to the second topic. The second topic was providing some sort of a central estimate and not just a BMDL. So this slide is an example from benzalphapyrine assessment in 2017, showing that as we calculate PODs, we are reporting the BMD along with the BMDL. So it is in the assessment document. And I gave a reference there to an example for an epidemiology study as well, fairly recently completed ethylene oxide assessment, where we provide both central estimates and lower bound estimates, or upper bound, depending on what the context is. So we think we are doing those already. The challenge is that it's not always easy to translate a central test estimate and a reference concentration or reference dose into a reference concentration, because there are uncertainties in going from one to the other. Which I'll come back to in a second. One of the other things we've been doing is presenting arrays of candidate toxicity values. And this can be from different studies. It can also be from different endpoints. And it allows a visual sense of to what extent are the conclusions based on a particular endpoint or a particular study, to what extent to different data sets get you to pretty much the same value. This is not a meta-regression in this case, but it's not unlike a forest plot in terms of showing different data sources and where those come out. So a couple things in terms of improving characterizing uncertainty. We're looking at model averaging. We're currently evaluating several methods for doing this. At this point, we're focused on dichotomous data. We think that will be easier to do. It's a more developed field. And looking at ways so that you can capture the different models would give you somewhat different estimates of BMDs. The other thing we're going to start doing in parallel with traditional RFC and RFD approaches is explore using an approach that the World Health Organization, the International Program on Chemical Safety, developed to characterize uncertainty not just in the point of departure, but in estimates of a risk-specific dose, much as Science and Decisions recommended that EPA should do. So that approach is basically a series of ratios instead of uncertainty factors treated as random variables, so derived from data and then applied in a probabilistic framework, generally assuming that the ratios are independent of each other and using the appropriate math, as was described actually in the NAS 2014 report, not describing this approach, but describing the mathematics of combining a series of factors. So what's interesting about this is you can go beyond just a sense of a central estimate and a lower bound on the point of departure to talk about a dose in humans and take into account uncertainty and extrapolating from subchronic to chronic or chronic to human exposures. With such an approach, you can then provide a variety of outputs. So that's a very simple mock-up on the bottom, just showing that you could, with a target dose, characterize a confidence interval with a central estimate and an upper and lower bound. You can also calculate distributions. You could also, there are a number of different central estimates you can look at. So it's very powerful if you can get to a point where you're describing distributions and not just picking one number. It also allows, in a situation where there's uncertainty, for you to both say, here's a dose below which we're quite sure there's not a risk, but here's also information about how uncertain that estimate is. Moving on to the third point of developing quantitative methods that will help with integration, we'll talk about a couple things, meta-analysis and Bayesian analysis. So meta-analysis is a tool, actually you saw an illustration from the TCE assessment, so it's not that we haven't used it before, but people are getting more comfortable with it. There's software tools that are available now that make it easier to do. And more we're trying to do this where we can. It's very powerful both in terms of helping for hazard characterization, because you can take a set of studies and come up with a more coherent combination of results, as well as for estimates of dose response. Bayesian approaches is something that's probably a little bit more exploratory. So one of the things we figured out is that to start getting more sophisticated in terms of probability, you also have to put your answers in terms of risks and probabilities. So the IPCS approach is not necessarily a Bayesian analysis, but it is very much a probabilistic analysis, which then puts you on a better footing for discussing Bayesian approaches. The approach we're looking at model averaging, as we've looked into it, it turns out that we think a Bayesian approach to the BMD models and then evaluation of the model averaging is going to be a better way to go than a non-Bayesian approach. And so we expect to be taking some of that forward into peer review within the next year. Within meta-analysis, there are a number of assessments where we've looked at Bayesian approaches to meta-analysis. I'm not sure those are public yet, but they've been worked on within the IRIS program. And there's some exploratory work. There was a poster last night that Leonid Kopilov had put together of using Bayesian networks. And the examples so far where it looks like it's useful is particularly integrating where you've got gaps in the data. And the particular example that Leonid produced was looking at a category where sometimes you've got human data, sometimes you've got animal data, but it's a mixed bag, and can you use Bayesian networks to basically integrate that? A related effort is that as we think about uncertainty analysis and providing central estimates and lower estimates, one of the audiences for central estimates are the economists within the agency who have to do benefits analysis. So we've got a memorandum of agreement with the Office of Policy and the Economic Analysis Group there, and trying to understand and crosswalk the two cultures so that the toxicologists are hearing from the economists what they need, and the economists are dealing with the toxicologists. There are some studies where we have epidemiology where it's fairly easy to provide a dose response estimate within a human population. In ethylene oxide, we were able to do it most clearly for occupational exposures because the epidemiology studies were occupational exposures. We want to look at the IPCS approach to see if it becomes a way to bridge that for animal data, whether we can get useful information from that that helps the economists do benefits analysis. So I don't think that was a recommendation in the 2014 report, but it relates to this issue of central estimates and understanding what central estimates would be useful to the user community. And now I'm going to ask Jason Lamberth to walk us through some of the new assessment methods.
Thank you. So I'm Jason Lambert, NCEA Cincinnati. What I'm going to share with you is really just a snapshot of a great deal of work that we've done in ORD and CEA over the past decade, primarily in response to two. One was a problem formulation related to our human health assessment mission. The other was with the publication of the National Academies report, Tox Testing in the 21st Century, A Vision and a Strategy, back in 2007. So the problem formulation faced with that's not called IRIS, we have other human health assessment lines, one being the provisional peer-reviewed toxicity values. We serve primarily Superfund. And what we were faced with when I joined the EPA back in 2005 was when we were tasked with evaluating chemicals of interest to Superfund. And what we found was when we went and scraped all the literature together, there was almost no or little available hazard and dose response information to inform a traditional human health assessment. So back in those days, unfortunately, we provided what was called a no value document. And truly, it was no value either to us or to our end user. So that was categorically unacceptable. So we decided, based upon what we learned from the 2007 report and this basic need for our friends in Superfund, was to come up with new ways to say something about hazard and dose response when you have a lack of data. So as you can see, I showed just a few of the reports that our group in NCEA have paid a lot of attention to, including just the recent National Academies report from last year using 21st century tox in health assessment. And so people have called it Tox 21, alternative methods testing, tox testing in 21st century. The newest moniker that I'm seeing across Europe and Canada and the US is we call it new approach methods in general. And so shifting the paradigm is been a continual process. It started with Bob Kavlock in the agency, leading us down this road with the formation of the National Center for Computational Toxicology, and then working closely with us in NCEA in the translational application of a lot of those new platforms and data coming out of their shop. And so in addition, we've worked with international partners and other federal partners. I've even worked with Sharon and Karen at P&G, since they live in Cincinnati, hometown crowd. They've done a lot of work, and much of what we've done has been based upon some of the framework, if you will, that our friends at P&G actually set up in the WU paper a long time ago. So the NAM toolbox, at least within our purview in ORD and CEA to date, and with our friends in the National Center for Computational Toxicology, includes the following. This is by no means comprehensive. It's just a snapshot of a lot of the things that we're looking at for application currently. And that includes data mining. And so the comprehensive collection and collation of existent data across the entire exposure to outcome continuum in a platform that you can use it in a modular way however you need it, but it's in a one-stop shop. Things like HERO is also another resource that we use. Chemoinformatics, this is not new. Structure activity's been around for decades. But the way in which we use it in human health assessment application is relatively new. And that includes structure activity, quantitative structure activity, and read across. And I'll share with you all in just a moment an example, read across examples that we use within human health assessment. High throughput exposure modeling. John Wambaugh and Peter Agegi and others have done tremendous work in trying to come up with exposure estimates where that data is lacking from empirical evidence. High throughput toxicokinetics. Long story short is there's even people in the crowd that have worked on this aspect. It's in vitro and in vivo modeling using reverse toxicokinetics to try and say something when you have cells in vitro. How can you translate that to an estimated maybe a human or animal exposure in vivo? Bioactivity. This is I use the word bioactivity, but this encompasses all of the omics, transcriptomics, proteomics, metabolomics, in vitro cell culture, bioactivity assays, and on, and even cell-free assays. Adverse outcome pathway, yeah, what can you say? Mode of action is still what we use, but adverse outcome pathway is the sexy thing to say right now. But really, it's just arranging your information from the exposure to outcome up to a population level in a manner which allows you to identify potential patterns or key events leading to an outcome of interest. And the manner in which we are investigating, I won't cover this today, but we're looking at how can you use the information that's less than apical or less than phenotype to inform both single chemical and mixtures of chemicals for assessment purposes. So today I'm going to touch on read across and some of our transcriptomics that we've done in an actual human health assessment application arena. So we use expert-driven read across, and again, we published a paper back in 2012. We were not the first people to come up with this, but we expanded on what was an evolving area of when you have chemicals that are like in areas like, I'm sorry if you all can't see this very well, but that Venn diagram shows that we use structural and physical chemical properties, we use toxicokinetic information, and we use available toxicity information. So again, think of this, we're in a data poor situation. So a chemical of interest that we're trying to evaluate has very little data, but we know its structure, we'll know some of the physical chemical properties, there might be some limited ADME data or toxicokinetic information, and the toxicity information, I don't want to overplay this, but it's typically things like LD50s, it might be eco species that we have information on that can inform toxicity. And so I'll say in our application, we use that type of information, but of the recent past, in the past year or two, we have expanded this to include less than apical information, so key event, MOA, AOP type bioactivity, to also use this as a comparator. The objective here is that you use this information not in a linear or sequential way. It doesn't have to pass some threshold to get to the next tier, but rather you use all of this information in a weight of evidence all together in a holistic way to come up with candidate sets of analogs that are similar to your target chemical of interest. This approach is commonly called many to one. And the reason being is when you assemble the candidate analog space, you then identify the single best analog within that space that can serve as the surrogate for your target chemical, both in terms of inferring or interpolating to the hazard of your target, and then using the single best surrogate as the basis for your dose response information for your target. And that is using, for instance, like a point of departure for the surrogate chemical as the basis for deriving a human health value for your target. I've expanded on this. When I say iris type, this is really any chemical that's not data poor, but for this meeting I call it iris type chemicals. So for chemicals that already have what we, potentially sufficient human and or animal information, the way we see this, the read across, same approach, just in a different problem formulation or compartmentalized way that you apply the information to fill data gaps, to augment weight of evidence, to provide, like Robin Ann said yesterday in her comments, to provide some of that mechanistic underpinning if you need it for your data-rich chemical, and potentially using some of that information to reduce some of your uncertainties. The category approach, this is what we call the poorest of the poor. So when you have chemicals that have almost no data of note useful for human health assessment, but we do know structure and we may know some of the physical chemical properties, whether they're empirical or estimated. Long story short is that you take your target and you try and find if it fits into a membership within an existing category or a category of analogs that you've assembled based upon primarily structural and physical chemical property aspects. The robustness of this approach is really dependent upon the density of your category, how many members do you have in that category, and it's highly reliant upon the weight of evidence supporting tox endpoints. Not meaning that your category, not all of them have to induce some liver endpoint or some thyroid endpoint, but rather at least it has to have some similarity within neighborhoods within your category that there is similarity based upon structure and physical properties. And the category approach assumes some close or similar adverse outcome or mode of action pathway. So for a category approach, we have used this in a human health assessment for extremely data poor chemicals again. We've filled the data gaps for hazard. We've extrapolated that hazard. The difference between expert-driven is you're interpolating hazard. For here, you're extrapolating the target chemical to a member within a category. So you're basically, your target has a nearest neighbor within that category, and you use the information from that nearest neighbor. For iris-type chemicals, again, data gap filling, augment weight of evidence, but one of the things I want to point out here is that third bullet, foundational member of a category. So it's a two-way information thing in a category approach. You can garner information from even more well-characterized members of a category for a data-rich chemical, but then that data-rich chemical can also then become a potential member of a neighborhood of that category. So we've used those approaches so far, not in iris yet. We're going to get to there. I want to move on real quickly to bioactivity. This is just a snapshot of some studies that we did with Rusty Thomas when he was at the Hamner Institute in North Carolina. Bottom line is what can you do when you have a situation, we don't have time to run 2-year rodent bioassays anymore, right? Well, can you run short-term in vivo animal assays, and how does the genotype-phenotype relationship play out as a function of dose and or time? And the graph, the regression there that I show you is just in a sample where we've done both non-cancer and cancer for a number of chemicals. And what we found, incredibly, 10 years ago, if you'd asked me what the dose relationship between genotype and phenotype was, at that time I would have said, several fold apart or order of magnitude apart, because you'd think that a biological pathway might be perturbed before you ever see some phenotypic expression of that. It's not true at all. It's actually over top, as a function of dose, virtually on top of each other. So we found close relationships between genotype and phenotype across oral and inhalation routes of exposure, rats and mice, and multiple target organs, liver, thyroid, kidney, both non-cancer and cancer. Question about in vitro. Yes, we're working on that. We're a little bit further off from that, but how can you use in vitro transcriptomics in a similar manner to inform hazard and or dose response? The things that need to get worked out with our friends in NCCT and elsewhere is optimizing metabolism protocols to how can a cell culture in vitro, how can we best make that relevant to what might be happening in an in vivo situation. And again, we would have to integrate the in vitro to in vivo extrapolation. I'm not going to spend much time on the GSCA. One of the big cautions or concerns that we received during scientific peer review of all of our studies in this domain was we use the lowest transcriptional pathway. Okay, so when you look at all the genes perturbed within a given pathway, let's say there's 30 genes, only half of them look like they were even relevant to the phenotype of interest. So a lot of people said, well, what do these top 5 gene perturbations have anything to do with the liver, because they were all brain proteins? So what we do now is we invoke gene set enrichment analysis to help better inform the relevant gene perturbations to the potential adverse health outcome of interest. So for data poor chemicals, we haven't applied this yet in a human health assessment situation, but we're close. The work is there, the underpinnings are there. We want to publish a little more on the GSEA approach. It was well received. We published last summer on this. So our vision is for data poor chemicals to use this type of information, short-term in vivo animal, or in the future in vitro, as the basis for hazard. We can also use this as an empirical basis for dose response based upon pathway perturbations, and eventually reduce the need for longer-term animal studies. For iris-type chemicals or data-rich chemicals, we could use this information again to augment weight of evidence like mode of action. This second bullet is something we've seen this, we just haven't published on it yet, but an opportunity to alert for off-target effects. So when you have a data-rich compound, you've collected all the information and you think you know all the hazards from human and or in vivo animal studies. When we've looked at the genotype or transcriptional pathway perturbations, there are perturbations that make you go, is there another target that hasn't been surveyed yet? And so that might lead to a futuristic approach where it's non-targeted analysis at first that leads you to maybe we need to interrogate a certain outcome. And again, potential for reducing uncertainties. My last slide. Working with Rusty Thomas and his group at NCCT, we're working on assembling a RapidTox dashboard. So all of what I just showed you and much more, the idea is to put this into a modular dashboard such that when you have a data poor situation, you might use the entire suite of what's available on the dashboard, and this includes all of the structure, phys-chem, bioactivity, transcriptomics, the read across, and much more, exposure. And in a data-rich situation, you could use it a la carte to fill your data gaps as needed. The idea is though to always use systematic weight of evidence, always be very clear with the intended audience of what this data is and is not, and the uncertainties inherent in it. And that's the future. So we'll continue on the data poor path and then grow our capabilities both within NCEA, with other federal partners and international partners. In academia.
So Jason got the great graphic here that, and no, George, you can't just push a button and have it all done.
We're trying.
We'll get there, right. So just to recap, Chapter 6 and 7 of 2014 made a couple recommendations. It's not the order I'd want them in, but that's okay. We are developing and applying quantitative tools. We're applying meta-analysis now. People feel like there are software packages that often allow them to do what they want. Where we need to develop more specialized tools, we will do that. We'll continue to expand the development of software for benchmark dose modeling. We talked about some of the things like model averaging and using some Bayesian approaches that are in the works. In terms of criteria for developing toxicity values, we are using those overall judgments about the integration of the confidence and the hazard to inform when we do a toxicity value, assuming the data allows us to do that. We are providing MLE estimates of BMDs along with the BMDLs in the document, and we are looking at ways that one might carry forward that characterization of uncertainty to human risk-specific doses rather than just stopping at the point of departure. And in terms of future directions, I think Jason gave you a good sense of some of the future directions with new assessment methods. And again, as we develop tools and experience with meta-analysis, probabilistic analysis, and Bayesian approaches, we'll continue to evolve. So I think we're done there. Thank you.
Okay, thank you. And those of you who have questions, remember to put your things up.